Students, today we will study development of microsporangia in Cycas. Each microsporangium develops from a single sporangial initial which is hypodermal in origin and not superficial. The sporangial initial arises in the hypodermis of the microsporophyll. It divides by periclinal wall into an outer primary wall and an inner archisporial cell. The primary wall cell by repeated periclinal and anticlinal division from a sporangial wall of several layers 1 to 6 thick beneath the epidermis. Some botanists designate that the sporangial wall cell as the endothecium and the epidermis external to the sporangium is exothecium. The cell of the epidermis external to the sporangium become thickened with the exception of two rows of cell along the adaxial root. They become elongated and mark the line of dehiscence. The archisporial cell undergoes division to form a mass of sporogenous cell. The sporogenous cell at the periphery of the mass form a single investing layer, the tapetum. The cell of the last cell generation of the sporogenous mass constitute the spore mother cell. They separate from one another, enlarge and round off and begin to divide. The tapetal cell along with the wall cell break down, providing nourishment for the developing spore mother cell. Each spore mother cell become a four-lobed structure. Its nucleus divides twice. The nuclear division is meiotic. Each haploid daughter nucleus migrate to one of the lobe of the spore mother cell. The lobe becomes separated by thickened partition. A single microspore develop within each lobe. Dehiscence of sporangium. It is longitudinal. When the microspore reach maturity, the male cone elongate considerably and rapidly. The scales separate from one another so that the sporangium are exposed. The sporangium loses water, and with the loss of water from its cell, the exothecium shrink. As a result, the spore wall split opens on the abaxial side along the elongated cell, making the line of dehiscence. The sporangium thus opened by a slit on its outer face. The spore fall down. The slits are radial to the center of the row. The liberated spores are dispersed by wind. Each spore is thickened at the opposite pole. The spore wall is differentiated into two coats. The outer is called exine and the inner is called intine. Thank you.